Good day, everyone. On behalf of Cambridge Health Tech Institute's Global Web Symposia series and our sponsor, Eurofins Discovery, I'd like to welcome you to Preclinical Secondary Pharmacology is an essential part of drug discovery. My name is Elizabeth Lamb, and I'm the host and moderator for today's event. Now, I'd like to introduce our speaker for today. He is Laszlo Urban, PhD, MD, Global Head of Preclinical Secondary Pharmacology and Preclinical Safety, Novartis Institutes for Biomedical Research. Welcome, Dr. Urban. The presenter ball is yours.
Good morning, good afternoon. This is Laszlo Urban from Novartis, Cambridge. I'm talking today about the preclinical secondary pharmacology and its essential part in drug discovery. The agenda first, I would like to describe what items we are going to discuss today. First of all, the definition of the preclinical secondary pharmacology, and then we will move into the profiling steps, how we set up for secondary pharmacology profiling, and how do we make an off-target risk identification and mitigation process. We will discuss step by step how we build up the models, for example, what kind of characteristics we have to look for. And then certainly we will talk about the interpretation and use of the secondary pharmacology data. And finally, we will provide a critical view and novel directions. The role of secondary pharmacology is very prominent during drug discovery phases. We have basically three pharmacology segments: the primary pharmacodynamic studies, which are concerned with the primary target, the therapeutic target, and the safety pharmacology studies, which are animal studies mostly relevant for submission of compounds for registration. In between these two, in red, is highlighted the secondary pharmacology studies, which is basically in vitro studies concerning off-target effects of clinical candidates or any compound during lead optimization. However, we have to consider that these three arms of pharmacology happen about the same time, particularly the first and the second one. But many times now in vivo pharmacology assays start very early on during the optimization phase. Our guidance is the S7A safety pharmacology studies for human pharmaceuticals, which was defined in 2001. But this is largely concerning the in vivo studies. What Novartis basically developed, and I will talk about the Novartis concept today, that we adopted this concept to a high throughput platform in a sense of profiling, obviously, and we introduced human assays at the early phase of drug discovery, which are determinant and also predictive for in vivo and clinical aspects of off-target activities. And we call it in vitro safety pharmacology profiling. The main difference between the two approaches that here we can mitigate on the chemical scaffold, while in the first phase, in the first paragraph, we manage, and mitigation is out of scope at that particular time. So just to highlight how secondary pharmacology became important for the pharmaceutical industry, this paper represents basically the approach of pharma and biotech companies to secondary pharmacology. On the left-hand side, you can see that there are in vitro pharmacological profilings which are generated for small molecules in most of the companies. And very few companies don't do this. It's almost 100% who oblige. However, there are differences where people use these capabilities, mostly for lead optimization, the green column, and development candidate analysis. But more and more people are using it to hit to lead, which is very important. On the right-hand side, you can see that it is represented in the regulatory submissions, and the data are generated both in-house and externally to a great extent. And it's very important that they align with the in vivo activities in most of the cases in the pharmaceutical companies. And finally, it's also very important that we get questions about secondary pharmacology data in submissions. So altogether, this slide shows that secondary pharmacology became an essential part of drug discovery. The regulatory environment is changing. in the middle of this slide I highlighted is the regulatory environment becoming more risk averse? And the answer is yes. And then secondary pharmacology is not currently mandatory safety study, which we include in submissions, but it might be the case. There are discussions about it. And this refers to FDA, but other authorities also consider secondary pharmacology. And I highlighted a Nature Reviews paper, which Nature Reviews drug discovery paper, which basically came from the FDA and gave an example where if we hit 5-HT2B, the serotonin receptor, in an agonist mode, and we know that this would cause fibrotic cardiac valvulopathy, then there is a possible regulatory action. As the last line in this shows, if no safety margin, then possible clinical hold due to serious and irreversible nature of the effect, which is quite serious. And as an insert shows here, it is, or it was, with the case of mediator, which is a fenfluramine-like molecule, and it caused lots of damage and suffering in patients, and particularly in women who used it as a slimming pill and caused cardiac valvulopathy. And in France, it is a major debate at the moment how to basically take care of this already made damage. When we look at the screening for secondary pharmacology, now we are going into the details. How to quantify safety risk is basically a quite tedious process. So first of all, we have to set up simple in vitro biochemical and cellular assays. And these assays concern basically off targets. We are not concerned about the primary therapeutic target and its associated adverse events, but the others, which could be membrane-bound, which could be intracellular, which could be nuclear. And keep in mind that in addition to the desired therapeutic effect, the secondary effects could be beneficial, deleterious, or neutral. And here's a case where we did some profiling for a compound, which is a COX inhibitor. So it shows the primary target lighting up in this sheet. But seems like all the other assays are pretty clean, except the beta-1 adrenergic receptor activity, which is mild. And if this is obtained during the lead optimization phase, one can basically do a series of compounds and find the best compound which has the least side effects, the least off-target effects. What remains is what is the translational value of the in vitro activity to clinical risk. That's what I try to elaborate on in the later slide. So what I will describe in the next couple of slides is a practical way to approach secondary pharmacology. And we will talk about here the Novartis way. So first of all, we need to determine target associations with defined or expected adverse drug reactions. And for this, we go for clinical evidence. This is the number one and most important. We basically try to identify targets which are associated with the most severe adverse events. The most important bit is to find drugs, for example, which are in the clinic, and we can define drug, target, ADR associations. The example I gave here is Celebrex, which is a fairly selective COX-2 inhibitor, and we can link this activity to cardiovascular, thrombotic effects, gastrointestinal bleeding, ulceration, and perforation. But in case we do not have any drug for a particular target, we use human genetics. For example, for the sodium channel NAV 1.7, which is involved in the transmission of pain and hyperalgesia. And if this gene is mutated, it could basically cause no feel of pain, no sensation of pain. So we would know what kind of effect we expect if we block this particular channel. If we don't have any clinical evidence, we try to rely on in vivo evidence. This is less important or has less weight than the clinical evidence, but still in some cases where we cannot rely on the clinical part, it becomes important. So here we consider knockout animal, CRISPR modulations, and some animal models which were developed copying particular diseases. So this panel basically shows you that we are focusing on the adverse events which are associated with particular known targets, and we are considering the most severity. Now, How do we do that? Basically, we would like to bring target and adverse events somehow close to each other. And in between is the genetics and the pharmacology. We use marketed drug testing, and also we use the OMIM. We use PubMed, OVID, and deep learning capabilities to do that. We use Pharmapendium at Novartis to search, for example, TERS, the FDA adverse event reporting system, and external databases in silico predictions. Now, if you watch the target and the ADR, if you all have these, it will change to hazard and risk. And we are now talking about how do we link hazard to risk. Hazard being the secondary pharmacology effect, and risk is what we see in the clinic according to the secondary pharmacology effects. The next question is, once we know what are our targets, to see how we can build a portfolio. Then we look to hit rates of targets. And this depends on experience. So, for example, if you look at the cardiac ion channels, then we know that in Novartis, we hit about 44% of compounds when we are examining hERG. And this hERG effect could be very, very weak, but it could be also very potent. Up to 30 micromole we test all of these assays. So in case of hERG, we have a high hit rate. For calcium and sodium, we have lower hit rates. Therefore, we basically design what is the volume for testing and what is the frequency. So we test for hERG weekly, but we do not do the same for the other two channels, and we test for hERG at a very high volume and much less for the other two to save money and also to have the high speed when necessary and the lessened speed if it's not. What I have to say also that in case of ion channels, when sodium and calcium channels are involved, we certainly allow higher testing rate. The next question is the quality and dynamic range of assays, the assay correlations. For example, here in this insert, you see the correlation between radioligand binding and Q-patch, which is the cellular hERG assay. You can see that between the two red lines, we have good correlation. But then above this line, we have lots of red dots. Every dot represents a compound. And what you see here that in the Q-patch assay, all of these compounds are negative, while in radioligand binding, some of them are even less than 1 micromole active. The reason why we keep them red is that they have very low permeability. And the cellular assay, therefore, cannot see them, as these compounds have to go through the membrane and then get into the pore of the channel. That's how they work. So one has to know this kind of thing, and then one can basically explain the discrepancy. The wide dynamic range is extremely important for profiling assays because we would like to have high confidence at a very high potency. So, for example, if a drug has an off-target effect at 10 nanomolar, it has to have high confidence. But another drug, which is tested in the same assay and has about 20 micromole potency, that has to be also the same high confidence. It's extremely important for the determination whether they would cause any side effects. The next question is basically the consideration of required modality, because side effects are modality dependent. So, for example, 5-HT2B has to be an agonist to cause harm. If it's an antagonist, we do not see much of target effect. The next panel is describing basically the logistics. So the testing protocols, when and how to test. We test human overwhelmingly and very rarely non-human in our assays. So these are transfected cells. Either they are biochemical assays, mostly radioligand binding, and cellular assays. The important thing is that we basically prevent side effects in the clinic. However, if we see discrepancies between our data and in vivo activities, we are able to do the same species in vitro and align with the observed in vivo data. Now, the next question is, should we use single doses or should we use dose response curves? And this is basically, according to any company, could be different. In our case, we start with dose response curves for two reasons. One is that this is cost effective, but more importantly, this gives a very quick result. So if you use a one single concentration determination in your panels, then you have to have a second run if you would like to have more precision. And after all, we are going basically to rank compounds during lead optimization. Therefore, we need precise data. We also need to align with phase, frequency, and volume, as I mentioned before. Then we have to consider individual targets or panels, target classes. So I'm defining basically the Novartis approach here. So we use general safety panels with broad target class cover because off-targets are not class specific. So we basically go by the rule I described before, the severity of adverse events and the frequency of hits. We use safety kinase panels for those particular projects which have a high hit in kinases, particularly the kinase-related projects. And we use the cardiac channel separately because it's largely electrophysiology related. The primary profiling basically will define follow-up in-house testing. So why we outsource panels, and it's fast and it's generating lots of information, once we fine-tune, we basically can run particular assays for mitigation in-house. I will give here one example for the human specificity, how important it is that we predict for the clinic and not just for in vivo experiments. So the case is related to the bile salt export pump, which is BSAP, and this emphasizes the importance of human data. The case is bosentan, which is for the indication of pulmonary arterial hypertension, and the target is the endothelin 81 receptor. It had a clear preclinical profile, but it had a hepatobiliary DILI, and it has a warning for risk of liver injury. What was measured that it has a fairly strong BSAP inhibition, but why was not it discovered in the rat in vivo studies? And here's an explanation from another publication. This apparently is troglitazone, but it describes that, for example, if we look at human hepatocytes, the enhancement of perchlorate in the cell due to different doses of troglitazone increase, while if we look in the rat, it remains flat. So you can see the huge difference that the human hepatocytes basically store the bile acids, while the rat does not. So that explains why this particular compound, bosentan, did not come positive in preclinical studies. And if we now know what is the exposure in the clinic, then the safety window would be only 40 instead of required 500 for this particular target when we consider free plasma concentration. So to summarize some information so far, we are basically looking for species-specific effect, poor prediction of animal models for abuse liability and suicidal ideation, for example, for VMAT2 inhibition. I will talk about it later a little bit. Functional changes in the absence of early morphological manifestation like PD3 inhibition, or drug-drug interactions due to CYP inhibition might be very different in the human environment. Toxicity developing at long latencies, 5-HT2B agonist-related cardiac valvulopathy takes months to develop. And finally, teratogenicity I mentioned here, which is related, for example, to bosentan endothelin agonism. Now, this brings us basically the general testing logistics, and this particular panel describes the Eurofins applications in discovery. You can see that the three different phases, the hit to lead, the lead optimization, and the candidate selection phases are basically aligned with hazard identification, elimination, and mechanistic understanding. This is hugely what all the pharmaceutical industry is following, and different panels are assigned to these different phases, which are basically fast. They require a small amount of compounds, and basically they deliver a capability to mitigate off-target effects at different phases of drug discovery. Now, the second slide here shows the Novartis way to go, which is very much or very similar, where we are using a core panel, which is here in this red elliptic. Principal panel, we call it, and we do it with Eurofins. It's about 30 targets, and we generate those response curves. And Novartis has a volume of about 1,000 compounds per year, which we test. Later on, or sometimes earlier, we use the comprehensive panel when we look at scaffolds, for example. Again, we are running it at Eurofins, and it's an additional 80 targets to the 30. and it has a single concentration at 10 micromolar, and it has limited access. And you can see that the impact is basically during first two phases is identification of potential off targets, then basically the mitigation phase, and finally, if we cannot get rid of totally of the off-target effects, we have some mechanistic studies to do. So here is basically the human targets associated with serious adverse events and high hit rate panels, the core panel and the broader panel. And then the later phase do not use anything specific, but basically analyze the data which were generated earlier. And finally, I would like to add that we seriously consider QSARs, SARs, and different mechanistic approaches in the artificial intelligence space, like using C, similarity ensemble association, and other tools. So we come to the data presentation. Here you can see a panel where a particular compound, in this case I used a clinical compound, which is an antipsychotic and hits many different targets in the principal panel. In our case, when we hover over the numbers, the data, then they will give basically the curve, and they will also give a description of that particular target and the associated adverse events. So you can see we have different panels like kinase panel and comprehensive panel, and if necessary, we look into different other areas. We have also a separate panel for cardiac safety, as it is of major importance. And we have a functional assay panel. As I mentioned, we can look at modalities. What I would like to emphasize also that we look at physicochemical properties, log-P, pKa, and also solubility, permeability, CYP inhibition, or CYP activation, which is very important in terms of analyzing these compounds together with the pharmacological data, particularly, for example, if we are looking for metabolites at later phase clinical candidates. Now, still talking about the assays, the third part is technology. So we basically aligned our assays so that we can use 10 millimolar DMSO solutions, which is the standard in the Novartis library. And for the panels, we use more than or less than 200 microliter. So we can afford it at very early phase. And we have a fast turnaround time within 2 weeks. I think it's a very important element to regularly benchmark the baseline. We find that in some cases we have shifts which are unacceptable. So in this case, for example, in the ion channel assay, amitriptyline is used as the reference compound, and we monitor it at every single assay set and in a timeline. Here you can see a couple of months timelines. And if it gets out of the load range, then we readjust. Now, what are the limitations here? Physical properties definitely play a big role. Solubility, permeability, lipophilicity, particularly when we are using, for example, plastic plates. Therefore, for example, with Q-patch, there are glass-covered plates, which give much better results. We look for aggregation and quenching. Now, it is important to use reliable assays. If they are not available, this is a big issue, then rather not to use them, because you can generate false data, which later on will hurt the project. If there is no SAR support for a particular off-target, still one can try, but it is not so easy. Once the SAR is established, I showcase this later, it's much easier to do mitigation. And certainly cost is an issue, how complex assays we can use and at what time. So early phase is still using the assays which are less expensive, fast, and highly reliable. So how can the in vitro safety pharmacology or secondary pharmacology help? I would like to demonstrate it very quickly on a PDE3 inhibition. We know that PDE3 inhibits pathways which are associated with positive inotropic cardiac effects. So they will increase contraction, strengthen the heart. And we know that we don't need it in congestive heart failure. So for example, Murinon, which is a marketed inhibitor, is withdrawn from use in congestive heart failure. But we have excellent SAR for PDE3, so we can basically look at different PDE3 effects. So what you see here on the left-hand side is basically a table of compounds from a real project. Every single row is one compound, and the scaffold is basically by Tanimoto. You can see in this red box, we have the same scaffold compounds. And you can see that they all have, or most of them have PDE3 inhibition. Once the project changed to different scaffolds, then they can get rid of this PDE3 effect. And they have basically two different choices here for two different scaffolds where this objective could be fulfilled. And basically we can test these compounds, and once they are negative in PDE3 and they still retain potency, then we can basically go ahead with this series. What I wanted to demonstrate here is, in a very simplified way, that once the project has a lead molecule, there are two options if we see off-target effects. Either the off-target effects, the potency of off-target effects tracks with the potency of the lead molecule, which is bad, because basically here it's very difficult to get rid of the off-target effect and the associated adverse event. However, if we can establish the way that we keep the primary potency of the compounds, but we can basically reduce the off-target potency, as you can see here along the green line, then we have a winner project. And here I need to talk a little bit about promiscuity. Pharmacological promiscuity in some cases is rampant. For example, in antipsychotics, molecules have many targets which they hit, and most of them are necessary, but they also bring in some side effects. And if you basically would like to remember one thing about pharmacological promiscuity, it is important to know that they are not a particular family-associated, pharmacological family-associated feature. So the message, if the primary therapeutic target, for example, is a GPCR, that does not mean that you only need to screen for selective agonists, other GPCRs. A little insert on the left-hand side shows that most of the cases when people are analyzing promiscuity, it is associated very clearly with log-P. The Novartis experience is slightly different, but en masse, we agree with this kind of approach, that it's very good to replace or reduce, not replace, log-P effects, then promiscuity would be lessened also. Now, the interpretation of secondary pharmacology is really paramount. So how do we make sense here? We can go back to Paracelsus and say poison is in everything and nothing is without poison. The dosage makes it either a poison or a remedy, which is hugely true. So the approach is this: secondary pharmacology data represent hazard only. We don't know what's the risk associated with it. Risk can be assigned only when the therapeutic exposure is defined. Specific organotoxicity is assigned to targets, and human-specific adverse events can be predicted by target engagement. So what we basically define is the safety margin for every single target or every single off-target in this case, which is most of the cases IC50, so inhibitory 50 effect, divided by the free effective plasma concentration. And this safety margin can be defined by target classes, and there are a couple of efforts here, for example, for GPCRs or some other targets. And for GPCRs, it was defined that at least the safety window has to be 10. So again, IC50 divided by the free effective plasma concentration. Or in the case of 0.3 to 3 times Ki is another guidance. What I like to demonstrate here on this particular slide is the integration of secondary pharmacology with the necessary risk assessment. So here you see basically three different antipsychotics: fluoxetine, aripiprazole, and risperidone. And we basically look at gynecomastia and hyperprolactinemia, which is not present in fluoxetine, and it seems like it's not present with aripiprazole, but it is a case of risperidone. And these data, what you are looking at, basically come from FAERS. FAERS is the FDA adverse event reporting system, which allows professionals and non-professionals as well to enter data. And you can see that the percentage of hyperprolactinemia in this patient population is very high. If we look at the D2 receptor, which we know is the reason for hyperprolactinemia, and you can see that the efficacy, or the IC50 here, the potency is 7.2 micromole for fluoxetine, but it's very potent in aripiprazole and in risperidone, which is basically in the low nanomolar range. But what makes the difference in between them? So we go down to the third arrow here, which basically describes the PK, the Cmax, the free Cmax in these three different drugs, which is 8.8, 1.3, and 15 nanomolar. It looks like risperidone has the most, but if we basically divide the D2 IC50 with the exposure, the 3 Cmax, then you can see that the therapeutic index or safety index is over 800 for fluoxetine, over 30 for aripiprazole. So these two compounds are safe for these particular side effects, but it's 0.71 for risperidone, which means that the side effect will manifest, which is according to the D2 effect. So this is the way we can basically look at marketed data and then define what kind of therapeutic index we need or what kind of safety window in the preclinical setting we can afford. The secondary pharmacology approach can be used also, as I mentioned, for something where we do not have proper animal experimental environment, for example, for suicidality risk assessment. And basically here we provided an analysis where we can look at basic assessment, the description by the FDA on particular targets is such that we have to test them if these targets are hit. If they are not, then we don't need assessment. But we have to look whether off-target effects with the same compounds exist. So we listed particular off-targets which are associated with suicidality. And if any of these targets are hit, then the answer is yes, we have to do assessment. If no, then we can go to the end and no assessment is required. But there is a third important element. So if we have yes to number 2, so we hit some of these targets, we have to see what's the CNS exposure. If there is no exposure, again, nothing required. If it's unknown or yes to the question that we have exposure in the CNS, which is a very low safety index, then we have to do the requirement. And I just highlighted some of these particular targets. For example, CB1, which is including suicidal tendencies, and rimonabant is the case in the clinic. This was withdrawn because of high suicidality. Now, looking at the determination of safety margin, I took one example, which is for PDE3 inhibitors. So here we have, as I mentioned before, very good SAR. We know exactly what kind of side effects we expect. We also know what kind of compounds are in the clinic, and we can determine basically the IC50, the free Cmax, the free AUC, and we know also whether they have a box warning or were withdrawn from the clinic. So once this is all taken together, then we can look, for example, at milrinone alone, and we can define the safety margin, which is about 1. But if you look at the other PDE3 inhibitors, they have a different safety margin. For example, vesnerinone is close to 8. So we basically define that under these conditions, we go always with the weakest link, and we define the safety margin at 10. On the left-hand side, you can see the adverse events which we recorded for this particular target. So how do we use it? And here is the same panel of compounds which I showed before, where we have PDE3 inhibition for a particular scaffold, and there is another scaffold which doesn't have this. So there are clinical candidates here, 3 of them, and the safety margins are very, very low, less than 10, which was proposed in the previous slide. And here you see the PDE3 inhibition is very potent. And with number 1 compound, we see degeneration in the cardiac muscle. With the other 2, we tested basically the LVEF and the heart rate. And in these echocardiography experiments, we got positive data. So we stopped these 3 compounds, and then we got one which has now over 50 safety margin, and this was selected to go to the clinic. So to basically consider all what we said in the previous slides, the most important thing is translation, 3 times or more. So how do we collect knowledge of target effects relationships? Obviously, literature mining, experimental work, target class knowledge, drug class knowledge, and all of this could be done by high intelligence artificial systems now. How do we define safety margins is the next question. Can we increase PK understanding? What is exactly what we need to define this? And do we have sufficient in vivo data? So I would like to demonstrate step-by-step determination of the safety margin for COX-2 enzyme inhibition. So here we go step by step. First of all, we identify drugs which target COX-2. So here we have celecoxib, valdecoxib, rofecoxib, and diclofenac, which is preferring COX-2. Then we identify potency in the in vitro assays, for example, in the Eurofins panel. And these are the IC50 values here next to the different drugs. The next step is to identify the recommended dosage. So here can use daily med or pharmapendium. And basically, you have to get to the right dose and then see what is the value, what kind of exposure we have in nanograms per mil. It's given most of the time. And all of this, what I demonstrate here, are FDA data. You have to be careful also whether it's fasted or non-fasted. So you have to find the right dose and the right condition together. Now, to be able to compare these drugs, we need the molarity. So we need to find the protein binding and the molecular weight of drug. Sometimes you don't find protein binding in human, but in most recent submissions, it's almost mandatory. Then we identify the hazard flags for the target. So we use FAIRS partially or clinical trials for these or labels. And this was taken from pharmapendium, and that represents the myocardial infarction. You can see that in comparison to the other ailment, this is a huge number, and a large percentage of patients might have some cardiac effect. So we can define basically what kind of adverse events we associate COX-2 with. And finally, we repeat it for various compounds. As I showed here, it was for valdecoxib, but now we go for the others. And then we identify common hazard terms and calculate the safety margin, which is in vitro IC50 divided by pre-Cmax, or if necessary, AUC. And this is how it looks like on the next slide, that here we list all the compounds which we analyzed, all the information on the potency, the exposure. We also identify here the side effects. And finally, we determine the the safety margin based on the information we collected, which comes to 10. And one can see here that this 10 is defined. So once we are dealing now with a COX-2 off-target effect, we have to go by these rules. Now, I just wanted to add a little bit more complexity, how it's not easy to understand all the ADRs. It's not trivial. So this case is about Biox, and the segment A shows the reporting by time, how many reports were entered into the FAIRS database. And B shows basically how these different timelines affect the accumulation of side effects. If you look at the abscissa, this is basically representing all the adverse events, and every single color represents basically one adverse event. You can see that there are 2 major adverse events in this case. One is the cerebrovascular incident and the myocardial infarction. And here in C, you can see that many of these reports were given by physicians, and it's a very steady flow. But when it became obvious that there was litigation possibilities, the lawyers joined in. So one has to look at with a pinch of salt. If we compare Vioxx, which was obviously withdrawn from the clinic, and there were lots of litigations because of these 2 side effects, Celebrex started out somewhat similarly, but because the input from the physicians was low, and that compares the two compounds clearly by the professionals, very first, basically the legal profession jumped on it and saw a particular possibility, which then basically evaporated at later stage. So one has to be very careful when we are looking at adverse reactions, particularly in the post-marketing phase. And finally, we mentioned the in vitro profiling substitute, or I wouldn't call it substitute, but helper, which is basically the IT considerations. We certainly can test only a couple of targets in comparison to the potential of targets, which could be thousands. We test maximum 200 to 300, and that's a little bit of overkill. So we use in silico approaches where we can test basically large number of molecules, and this is the true scale of promiscuity which we can reveal. I mentioned the similarity ensemble association method, but there are many others which different companies are using. So in conclusion, target-based hazard identification is a major component of inexpensive, fast and successful integrated risk assessment and mitigation at a very early phase of drug discovery. The concept binds together three major components: the ligand, target protein, and adverse drug reaction. And certainly, this triad is very important to maintain. The translational value of the concept is significantly enhanced by using PK/PD information, as I demonstrated. And the combination of experimental and in silico approaches basically expand the scope. The final assessment is basically that early safety and assessment and mitigation is guided by high confidence reverse translation. It identifies hazards associated with scaffolds and molecules, contributes to mitigation of adverse events and risk assessment of drug candidates. It also provides measure of selectivity, promiscuity, detects invisible adverse events which develop during chronic in vivo or clinical exposure of the drug, and also detects human-specific effects not manifesting in regulatory in vivo pharmacology assays, and guides preclinical and clinical safety assessment for management. However, there is always a however, never use in vitro data without context of expected or measured exposure. They are usually, most of the time, meaningless without these dimensions. Disease pathomechanism might influence ADR profile. For example, with diabetic patients, hERG is more sensible. It could be more effective. hERG inhibition could come through, while in a non-diabetic patient population would not. Beware of overemphasis of importance or underestimation of safety windows. Only serves the small molecular entity drug discovery, not applicable to biologics and gene therapy. However, the profiling concept remains valid for these areas. And please remember the metabolites, because they might, particularly in case when there was serious mitigation, they might come back with a major metabolite. And that comes to the acknowledgements. I would like to acknowledge many, many people who worked on these projects during the past 15 years when we developed the secondary pharmacology. Finally, I would like to provide the reading material, and thanks very much for listening to this presentation. Thank you.
Thank you very much, Dr. Urban. Our first question for you: does the determination of an off-target safety window depend on a particular assay?
Okay, thanks very much for the first question. Yes, it does. And this is a really important matter, because once we determine the safety window or safety margin, whichever way we call it, we use a particular assay for the drugs which are already in the clinic. So it is essential to use the same assay for the clinical candidates or for the lead optimization phase molecules, because there is a fairly large variation in between the assays. So for example, if you are testing compounds with Eurofins and you would like to compare those data to drug effects, one can basically determine in the same assay the efficacy of the clinical compounds or apply for information which exists already in bioprint and basically use those data together. But I think the message is really that it is an absolutely essential part that you compare apples to apples, because otherwise the variation could be large. And this is doable these days, so I don't see it's a big issue. However, the other element here, which is the PK data, most of the time when you are looking, for example, for the marketed drugs, you rely on published data. And I strongly encourage you to look for the FDA data and not always the otherwise published data, because then you have a fairly high standard. I can see also another question coming up, which asks about the in silico predictions. Yes, I would, but that takes a little bit more time. What I can say here in a nutshell, that we are using various aspects of in silico determination. At the very early phase, we have a couple of QSARs, which we use once we accumulate some data on particular off-targets. We use it regularly for hERG, for CYP inhibition, but also for some GPCRs and some of the most frequently hit kinases. What we generally use for trying to find off-target effects, regardless of a particular space in pharmacology, is the SEA, the Similarity Ensemble Association, which was developed originally by Brian Scheukert's lab at UCSF, and I have very good experience with these. And C-Change is basically the company which provides this information. They are also working with the FDA. However, there are many other assays applications people use. So if you have a good random forest model and you apply it to reverse translation, you can achieve a fairly good predictivity. Just one more sentence. If you'd like to learn more about our approach, there is a Nature paper in 2012, which I cited at the very end, and that describes the model and the methodology we are using.
Ah, perfect. Our next question, what is the advantage of secondary pharmacology profiling in comparison to gene expression profiling?
In this case, I would like to say up front that one doesn't replace the other. They are used in parallel in our case, and I believe in most of the pharma and biotech research, because they address different things. The reason I was emphasizing the secondary pharmacology approach, because basically we are looking for proteins and ligands. And after all, some of the gene expression profile does not necessarily translate into proteins. So one has to do both. I think the gene profiling guides us to the great extent. If we can apply it early on, then we can go after the proteins which are associated, and we can even develop assays if it's absolutely necessary and look at the affinity of the compounds. But basically what we try to do, and maybe I didn't emphasize it to the greatest extent, that we have to rank molecules when we are doing the lead optimization phase. So if we have reliable affinity-based assays, then we can basically rank the molecules by the different off-targets and certainly by the primary therapeutic affinity.
Thank you. And we do have one more question. Why not use alternative profiling assays available on large-scale protein arrays?
I think this question was asked many times, and we have a couple of CROs where these are available. I do not want to scare people away from this, because in particular cases, we are using for lead optimization phase this kind of assays. However, the problem we were facing that many of these assays require a large volume of protein, so the price is much higher. And while we have highly reliable radioligand binding or cellular assays which come in a kit and it's relatively inexpensive, we prefer to do this. And only in cases where we need a higher volume, very specific profiling effort for a particular project do we apply this. It's nothing wrong with these assays. Many of the commercially available assays have high reliability, The only reason that for this regular profiling, which we provide en masse for Novartis projects, this would be too expensive.
Excellent. Thank you so much. That takes care of all of the questions. So I would like to take this opportunity to thank our presenter for today, Dr. Laszlo Urban. Thank you so very much. I would like to thank our sponsor, Eurofins Discovery. Most of all, I'd like to thank all of you who came and spent this time with us. We very much appreciate you spending that time with us. We know you're very busy, and we hope that it was a pleasant time and one that gave you some answers to your research questions. So with that, on behalf of Cambridge Health Tech Institute, CHI, I'd like to thank you so much for attending and have a great day.